Is gain-of-function research a biohazard? In 2020, a novel coronavirus showed up 20 kilometers away from what I'll call the Wuhan Institute of Novel Coronaviruses, leading some academics to conclude there might have been a lab leak. But government-funded media immediately said a lab leak was an insane conspiracy theory. And Chinese authorities, who are known for transparency and honesty, concurred with this assessment. But the CDC director threw shade at that narrative and said it was probably a lab leak. Meanwhile, the government protected citizens by banning 8-year-olds from the beach, and society got a little weird. But the thing that received shockingly little press is whether or not we should fund something called gain-of-function research, a technique where scientists make viruses that are normally harmless to humans into the deadliest pathogens on the planet. But why are a small group of unelected scientists creating biological weapons of mass destruction in their laboratories? And is it possible that this is the worst idea of all time? I did a lot of research on my computer, and what I'm about to tell you might be somewhat alarming. There are some arguments over the definition of gain-of-function, but broadly, it's any modification that enhances an organism. Wearing glasses makes your eyes gain function. Taking steroids makes athletes gain function. And CAR T therapy, a technique used to fight cancer, causes the immune system to gain function. But this isn't what the public is concerned about, and the narrow definition of gain of function is basically research that makes viruses more dangerous to humans. And in 2012, scientists created a virus that was so dangerous that reviewers initially refused to publish their paper, out of concern that terrorists could use the paper's findings to kill everyone. But the paper was eventually published, and one of the reviewers calmed the fears of the public by stating, "I can't think of another pathogenic organism that is as scary as this one. I don't think anthrax is scary at all compared to this," which made me feel much better. There are lots of ways to do gain of function, but the researchers did it using a technique called serial passage, which is when one animal is infected and then placed with other animals so that natural selection can modify the virus's properties over time. It's basically the opposite of being put in quarantine. Scientists had weaponized evolution to create biological WMDs. So why are we taking these risks? In order to beat your enemy, you have to understand your enemy. But viruses mutate so much that they become hidden from our immune system. The good news is that not all mutations are possible. Some parts of viruses are so important that modifications to them can result in an evolutionary dead end. These parts are said to be conserved. For example, when viruses multiply, they need help from something called a protease. The protease acts like scissors and cuts long proteins into smaller parts. We know this is true because this is an actual image of a protease for SARS-CoV-19, and this is the part of the scissors that does the actual cutting. And this is an antiviral drug. It binds to the scissor and prevents it from cutting. The result is that viruses can't multiply. By attacking these conserved parts of the virus, we have more confidence that it will be difficult for the virus to mutate and evade us by creating different versions of itself. And we can discover more weaknesses in the virus. Nature is already an uncontained virology lab. And because humans and animals are so close together, it's a guarantee that more outbreaks will emerge. And somehow, we need to be prepared for this. The previously mentioned 2012 study focused on a bird flu virus that is a nightmare fuel to virologists. At the time, we only knew bird flu can spread from animal to human. The study appeared to prove that if the virus mutates enough, it can gain function and spread from human to human, which is scary because bird flu has a 60% mortality rate. With the viral genes we discovered in this study, health workers might be able to perform genomic surveillance in nature. And hypothetically, we can predict what vaccines and antivirals we will need if the virus appears in humans. But can playing God ever backfire? Humans have a pretty bad track record with weapons of mass destruction. The US government has repeatedly used chemical and biological weapons on its own citizens. Occasionally, we find incredibly deadly pathogens just hanging out in places we aren't supposed to. Laboratories aren't always 100% locked down, and the government is currently missing 6 nuclear weapons, which I empathize with because I've accidentally left my phone at the bar a few times. And it's not just accidents or government terrorism that we have to be careful of. We have to be concerned about regular, old-fashioned terrorism. In 2001, letters full of anthrax killed 5 people. And the way the letters were written made law enforcement somewhat suspicious that maybe this wasn't an accident. It turns out that the anthrax came from a BSL-4 laboratory, which is our most secure type of lab. And eventually, the FBI said letters were sent by a researcher named Bruce Ivins who happened to have a patent on anthrax vaccine development. This whole story is very strange because this researcher also happened to be advising the FBI on how to investigate this case. Just to reiterate, we seem to be having some difficulty with lab safety. This is a non-comprehensive list of biosecurity incidents over the last 25 years, and it includes the 2004 lab leak of a coronavirus. You heard that correctly, a coronavirus killed people after being leaked from a Chinese lab in 2004, which is kind of scary. And the argument for developing antivirals and vaccines before a pandemic is no longer so strong. Research now moves rapidly, meaning that needing time to prepare for pandemics might not be as important. This is COVID-19. We discovered its structure within 10 days, and within 3 days of the publication of this structure, we produced our first vaccine candidate. This has led to some people saying gain of function is like searching for a gas leak with a lit match. In other words, it illuminates very little, but the risks are extraordinary. We are taking viruses from remote places in nature that humans might never go to and placing them in labs and dense urban environments. Then we are turning them into superviruses that might never ever occur naturally. If nature is a deck of cards, we have given it a bunch of aces. Scientists who run these experiments are making a species-wide decision without much feedback from the people they could potentially cause harm to. And if these superviruses escape, then we are all effectively taking part in a clinical trial that we did not consent to. And this might be why a new executive order on gain of function was issued in 2025. Specifically, the government has instructed itself to develop new gain-of-function research regulations, and you will no longer be allowed to use U.S. taxpayer dollars to fund gain-of-function research in specific countries like China and Iran, which is controversial, but some scientists have been asking for this for years. Do you think this is a good idea? Let me know what you think in the comments. And remember to like and subscribe to support me.